We have the concept of the webinar today is that I will give you first a more broad introduction about mechanistic foundations of tES, and 3 of my students will then give you some information about newly available protocols, so intensified protocols, but also some qualitatively new stuff, especially for stimulation of oscillations. So for mechanistic foundation of transcranial electrical stimulation, this belongs to the family of non-invasive brain stimulation, which includes mainly TMS and transcranial electrical stimulation. A couple of techniques available. We can use activity in the brain with TMS. We can induce plasticity with rTMS, repetitive TMS, with paired associative stimulation, pairing peripheral nerve stimulation, with brain stimulation, but also with transcranial current stimulation, so one of the topics today. And we can also modulate oscillatory brain activity with transcranial alternating and random noise stimulation. So what's important to know is that in difference to TMS, the primary mechanism of action of tES is that you don't induce action potentials with strong currents, but you have a relatively weak electrical stimulus, which is not able to induce activity, but to modulate resting membrane potentials. And you can see this here, that that's a rationale of the techniques that dependent on the direction of current flow in relation to neuronal orientation. You will depolarize or hyperpolarize resting membrane potentials, not up to the threshold. This makes it more or less probable that incoming activity results at the end in an action potential, and this incoming activity makes it over this action potential induction threshold. I show here the effects on the whole neuron. It's also important to know that at the whole neuron level, current has to flow always in and out of a neuron, so you will have de- and hyperpolarizing effects on different parts of the neuron, and you get relatively uniform effects with standard protocols because the axon and the soma of the neuron has more ion channels, more receptors, so it's more sensitive to the amount of stimulation. So what defines the effects of the stimulation is more this area than the apical dendrites. That tES has effects on cortical excitability and activity has already been shown in animal experiments in the 1950s and 60s. It's a study here by Bindman and colleagues conducted in London, where you can see that during tDCS, you get, compared to baseline activity, you get with a cathode over the target area. This was a somatosensory sensory motor cortex. You get a reduction of spontaneous activity. And with the anode over your target area, you get an enhancement of activity. You can see that this is spontaneous activity because it looks quite chaotic. So you have groups of spikes, groups of action potentials, and you have also some breaks here. So this looks quite spontaneous. Well, these are effects you get during stimulation. The important thing for us to take this up in humans was that when you stimulate for a longer time, you get neuroplastic after effects of the stimulation. So you can see here that with the cathode over the target area, that's spontaneous activity before stimulation. Then you can see here the stimulation, and after stimulation, you can see that the spontaneous activity is largely reduced, and this effect lasts for more than 1 hour after the end of stimulation. And you put the anode over your target area, you get antagonistic effects. So here, 2 stimulations, and you can see that compared to baseline activity, there's enhancement of activity, which in this case lasted for more than 5 hours after the end of stimulation. So that's animal experiments with the electrodes directly on the cortex or in the cortex. We transferred this to experiments in humans. So that's kind of our standard protocol we started with. Our reference area was a motor cortex. We have here return electrode. You can see that the electrodes are relatively big, so you don't get a very focused stimulation. You can also see that this is not really rocket science, so relatively simple stimulators. Important thing is here that you get constant, that you have a constant current stimulator, not a constant voltage stimulator. Similar as in the experiments with rats, we get polarity-specific effects with short stimulation. which don't induce after effects. So especially for the electrode position here, with the target electrode over the motor cortex, the other electrode contralaterally over the supraorbital area, you get an excitability enhancement measured via motor evoked potential amplitudes induced by TMS, excitability enhancement with the anode over your target area, and excitability reduction with cathode over your target area, stimulated here for 4 seconds. This is too short to induce after effects. But if you stimulate for a longer time, so we stimulated here between 5 and 13 minutes with the anode over the target area, again motor cortex, you can see that with 5 and 7 minutes stimulation, you get an enhancement of cortical excitability, which however lasts relatively shortly, so only for a few minutes. And you stimulate for a longer time, you get longer lasting excitability enhancement, lasting here for about 60 minutes after the end of stimulation. And you convert the polarity of the stimulation with the cathode over the motor cortex, you get antagonistic effects. Here also relatively short lasting stimulation with 5 and 7 minutes stimulation, longer lasting effects for up to 1 hour with 9 minutes stimulation. With respect to mechanisms, we speculated that this might be glutamatergic plasticity, from which we know that depolarization of neuronal membranes activates NMDA receptors, which are then furthermore activated, of course, via glutamate release, and this enhances calcium concentration in your neurons. Dependent on the amount of calcium concentration, this results in insertion or removal of AMPA receptors to or from the subsynaptic membrane. AMPA receptors work as ion channels, so they're conductive for sodium, and you can imagine that with a similar amount of glutamate in the synaptic cleft, the more of these receptors you have, the higher the probability that you get an action potential via glutamate release, and the less receptors they have, the less you have less probability to generate an action potential. So that's in a nutshell how plasticity of glutamatergic synapses works. You can see also here, I won't get too much into detail here, that GABA modulates glutamatergic plasticity, and that also a couple of neuromodulators is involved and has a modulatory effect on plasticity, like dopamine, acetylcholine, serotonin, also adrenaline, noradrenaline. Won't get too much into details here. We explored the dependency of tDCS-induced plasticity from the glutamatergic system by pharmacological experiments, and we blocked NMDA receptors. You can see here, as compared to placebo medication, you don't get any plasticity anymore by anodal or cathodal tDCS. So glutamate and the NMDA receptor is critically involved. This was also shown by another study where we enhanced activity of the NMDA receptor by D-cycloserine. You can see here there's an enhancement of the duration of the after effects of anodal tDCS. GABA is also involved in the after effects of tDCS. So we did here first study again motor cortex with TMS, where we saw relatively minor effects of GABA enhancement with lorazepam on tDCS-induced after effects. So you can see here slight delay of plasticity induction under lorazepam as compared to placebo condition, but no really clear effect on cathodal tDCS-induced plasticity. A solution for this bit difficult to interpret results came a few years later by a study conducted in Charlie Stack's lab in Oxford, where they did a magnetic resonance spectroscopy study and showed that independent from stimulation polarity, there was a reduction of GABA, of the amount of GABA in the motor cortex after tDCS. And this means inhibition is reduced. So reduction of GABA has probably a kind of gating function on tDCS-induced plasticity. So to put together, after effects of tDCS depend on glutamate. GABA reduction might also contribute. For tDCS, calcium-dependent glutamatergic plasticity can be assumed, and neuromodulators affect the strength and direction of tDCS effects. Okay, these were so far. This is so far for the regional effects of tDCS at the cellular level. And what we could also show is that tDCS induces network effects. And that's also important for the mechanisms of actions of the stimulation technique. Rafael Polanyi, I want to show you two studies he conducted a few years ago. Here the fMRI study, where he did first resting state MRI, then 10 minutes real or sham tDCS, and then again 5 minutes resting state MRI to look for network effects of tDCS. And what he showed is in a seed-based analysis, when he puts the seed over the motor cortex, and you can see that there's enhanced connectivity between the motor cortex and premotor and posterior parietal areas, so important cortical parts of the motor network. And in a consecutive EEG study, where we did resting state and motor task related EEG before and after 10 minutes real or sham anodal tDCS, he could show that there's a specific effect on task-related functional connectivity as obtained by EEG. So the motor task was finger tapping, and when we compared finger tapping before and after real tDCS, we could show that there's enhanced activity in a relatively widespread cortical network, but with the strongest connectivity in motor-related areas. Of course, this can be we can get to this result because of repetitive finger tapping or the effect of tDCS in both. So here we contrasted the connectivity after real and sham stimulation. And you can see here, important thing here is that the pattern of enhanced functional connectivity in the gamma frequency range is similar as that you can see here, but it's a bit more restricted. And what this shows you is that tDCS does not induce some random functional connectivity, but that it's somehow a task-related network which is activated. So it's a synergistic effect of task performance-related activation and tDCS effect that strengthens this network. So functional MRI and EEG allow identification of stimulation-induced alterations of functional connectivity of interregional cortical networks. That's so far basics of tDCS in a nutshell. Want to make a few statements about oscillatory stimulation with alternating currents. Stimulation intensity is similar as with tDCS, so relatively low level stimulation. The difference here is that we have sinusoidal stimulation. And in the first study, which was conducted by Andrea Antal and colleagues, they looked for neoplastic effects of this sinusoidal stimulation with alternating currents. With different frequency bands within the physiological frequency range of tDCS, and you can see here that they didn't get neoplastic effects of the stimulation. So the primary effect seems to be somehow different, but this doesn't mean that the stimulation is inefficient, but there were stimulation frequency dependent functional effects. They showed this for motor learning tasks where subjects learned to sequence. Sequence learning is shown by reduced reaction time during learning. You can see here that with 10 Hz stimulation, it's known that 10 Hz frequency is important in the motor cortex for performance of this task. There's a reduction of reaction time, so improvement of performance of the sequence. When we had here a random order of stimuli, you don't see any effects. You have this functional effect. Another interesting study showed in the visual cortex that effects of the stimulation depend on spontaneous frequency. So when stimulation was done during light conditions where beta frequency is dominant in the visual cortex, you see a reduction of phosphine thresholds only with beta frequency stimulation, whereas in the dark where alpha frequency is dominant, you see a reduction of thresholds only under alpha frequency stimulation. So this shows that tDCS does not induce in the conventional protocols oscillatory activity, but it strengthens or entrains already dominant activity. In a recent study, we were interested in mechanistic effects of the stimulation. Miles Wisniewski conducted a study in our laboratory where he did beta frequency stimulation over the motor cortex, and we combined the stimulation with dextromethorphan, which blocks NMDA receptors, because we wanted to learn about the contribution of the glutamatergic system on this oscillatory modulation via tACS, 20 Hertz stimulation. And what he showed is that this stimulation resulted in an enhancement of oscillation in the beta frequency range with 20 Hertz, which was lasting for up to 60 minutes after the end of stimulation. Interestingly, when we applied dextromethorphan, so we blocked NMDA receptors, we didn't see this effect anymore. And this effect was also not completely restricted to the stimulation area, but you can see here also that there was enhanced connectivity in the primary motor and posterior parietal region, also relatively long stable, not as but a bit variable. You see this also here, standard error was relatively big. That is also dependent on NMDA receptor activity. If we did block this, we didn't see this anymore. We additionally did TMS afterwards, motor evoked potential amplitudes over the motor cortex, and saw that we didn't only enhance oscillatory activity, but we did also enhance plasticity. So we did induce plasticity. Enhanced motor evoked potentials were also present after 60 minutes after the end of stimulation. These were also dependent on NMDA receptors to block and abolish these effects. So we achieved alteration of oscillations via prolonged tDCS. These effects were frequency specific. We saw also an enhancement of synchronization with neighbored areas. We had relatively regional effects, but this was a 4x1 electrode montage, so a bit more assumed, a bit more focused stimulation. It had additional neuroplastic effects, and both the oscillatory and neuroplastic effects depended on NMDA receptors. So I could show you here that tDCS entrains oscillatory cortical activity. It has a modulatory but not an inducing effect, which is important, I think. And for most frequencies, neuroplasticity is not induced with standard protocols, but with enhanced protocols. So enhanced intensity, enhanced duration, you get, at least with certain frequency bands, you get also neuroplastic effects. So to finish up in general, transcranial electrical stimulation induces acute alterations of cortical excitability and activity. Prolonged tDCS induces neuroplastic after effects. tDCS entrains cortical oscillations. Some frequencies induce also neuroplasticity. And beyond regional effects, I think that's also important to get also network effects. Some limitations are that the effects are often moderate, and effects are mostly modulatory and thus dependent on brain state. And we tackled these limitations with the studies my students will show you in the following. So that's it. Many thanks for your attention, and that's the group in bad mood this time.
Thank you very much, Michael. What we'll do now is, I think we can go directly into the next presentation and save the questions until the end, so a discussion at the end. So next up we have Desmond Agboada from Michael's group, who will now take over the presentation screen.
Okay. All right, so can you hear me?
Yeah, all good, Desmond, and I can see the screen as well, your slides, so we're good to go.
Perfect. So I'll be presenting some part of my PhD work, and it's titled Optimizing tDCS by Modification of Stimulation Parameters. So as Michael already explained, so tDCS is a non-invasive brain stimulation technique, we all know. And in the motor cortex, with the anode placed in the primary motor cortex, we have anodal stimulation, and with the cathode placed over the target region, so or the motor cortex, the cathodal stimulation. In one of the earliest tDCS studies, it was shown that the parameters of stimulation, that is intensity and duration, linearly correlate with the induced after effects. So in one of the earliest titration studies, if you see the figure to the right, you see the lower intensity of 0.2 milliampere induce the lowest effect, whereas the highest intensity of 1 milliampere induce the highest effect. Likewise, for the duration as well, there was some form of linearity with duration and induced effect of tDCS. There's a little bit of background noise. Okay. All right, so I'll continue. So, as I was saying, in the motor cortex with TMS-induced MEPs, we can see enhancement of cortical excitability with another tDCS, and then a reverse, so a diminution of cortical excitability with cathodal tDCS. Now, the effects of tDCS, as Michael showed earlier, can outlast for longer period of time, depending on the duration of stimulation and, of course, the intensity as well. And as it was shown in one of the earlier studies, if you stimulate between 5 and 13 minutes with 1 milliampere of another tDCS, you can see enhanced after effects that last up to about 1.5 hours after stimulation. But over the last few years, we have seen that the after effects of tDCS is actually partially nonlinear with respect to intensity in cathodal stimulation, and then with respect to duration with anodal stimulation. So with cathodal tDCS, Georgi Bachiskadze showed in 2013 that when you increase the intensity of cathodal tDCS from 1 to 2 milliamperes with 20 minutes of stimulation, there is a conversion of the direction of after effects from a diminution to an enhancement. That is the picture A. And with another tDCS recently, Asif Jamil showed that within a parameter space of 0.5 to 2 milliamperes, the effects at the group level of these 4 different intensities are similar. And on the right-hand side, the picture C and D, Montesilvia showed that with 1 milliampere of another tDCS stimulation with 30 minutes, we see an enhancement of after effects. But when you prolong the duration of stimulation from 30 minutes to 26 minutes, so continuous stimulation, instead of an enhancement, there is a reversal of the direction of after effects. So you see a diminution of excitability. So this nonlinearity that has been shown recently has led to a lot of thinking as to what is the basis of this nonlinearity and how this nonlinearity affects the application of tDCS in cognitive and clinical studies. And of course, how best do we optimize the tDCS parameters so to know exactly at which point does the stimulation convert from enhancement to a diminution. So in cognitive studies, and most importantly in clinical studies, we need to be sure about the efficacy of our stimulation protocols. And recent reviews in some clinical studies have shown low efficacy of some tDCS parameters. And so this has called for a more systematic approach to expand the current parameter space of tDCS with respect to stimulation intensity and duration to find the most optimal stimulation protocols. So in one of our first optimization studies, we aimed to expand the conventional parameter space, so which is normally between 1 and 2 milliampere, and a stimulation duration of between 10 to 20 minutes. We aim to increase the intensity to 3 milliampere and then the duration to about 30 minutes to see if we can enhance efficacy. So we titrated this expanded parameter space, and this was the experimental procedure. We first measured baseline cortical excitability with TMS-induced MEPs, and then we applied our intervention, which is a combination of the 3 current intensities, 1, 2, and 3 milliampere, and then 3 durations, 15, 20, and 30 minutes, and compared these protocol combinations with sham, which was 1 milliampere stimulation for 15 seconds. Immediately after the stimulation, we measured after effects every 5 minutes up to 30 minutes, and then every half an hour up to 2 hours. Same day evening, next day morning, next day noon, and next day evening. So a long period of after measurements. So we found that, so these are the results, A, B, and C. So on the vertical axis is your normalized MEPs, and on horizontal axis you see the duration of the after measures. And then you can see a baseline, the dotted line drawn from 1 millivolt is the baseline cortical excitability. You can see for 1, 2, and 3 milliamperes, the after effects was stable mostly after 2 hours, and then it came down to baseline for almost all of them. So we see an enhancement of after effects as has been reported in previous studies. But we did not see any significant difference between protocols of 1 and 2 milliamperes or stimulation durations of 15, 20, and 30 minutes. What we observed was a slight enhancement of 3 milliampere protocols of 3 milliampere, which was not significant, but it was a little bit of it was a trend-wise effect where the higher intensity or the more intense stimulation showed slightly better cortical excitability. The effects, however, were still not in the late phase LTP-like plasticity range. So we were not able to induce very long plasticity. For most of the protocols, we were able to induce about 2 hours or a little bit more. So we were still in the early phase LTP-like plasticity. So for us, optimizing tDCS is ability to induce a longer lasting stable plasticity. So if we cannot induce this with early phase LTP-like plasticity with single stimulation, we have to think about other ways of inducing late phase LTP-like plasticity. And one way of inducing LLTP is by repeated stimulation. With this protocol, you are stimulating a number of times with a critical interstimulation interval. And your ability to induce late phase plasticity is critically dependent on these intervals between the stimulation. Now, this has been shown, as Michael showed earlier, this has been shown back in the 60s, where in the rat cortex, application of weak positive direct current elevated spontaneous firing in the cortex and increased cortical excitability. And when a second application of this direct current was applied, there was an even longer elevation of cortical excitability, which shows the relevance of space stimulation and how useful it can be to induce late phase effects. In humans, the first study to use this space stimulation technique to induce late phase LTP-like plasticity was the Montefiore study in 2013. With no interval, when 1 milliampere 30 minutes of anodal protocol was applied in the motor cortex, there was, as we expected, an enhancement of cortical excitability that lasted up to 1 hour, which represents an early phase LTP-like plasticity. When the stimulation duration was prolonged from 30 minutes to 26 minutes, there was a conversion of the enhancement to a diminution, which is LTD-like plasticity. When an interval was introduced between the first and second stimulation, you can see there was an elevation of cortical excitability that came down to baseline after 2 hours, but from the next day onwards, there was an increase in cortical excitability, which shows a longer lasting after effects. But when the duration or when the interval between the first and second stimulation was very long, so it was 3 hours or 24 hours, there was no enhancement of cortical excitability, which shows here that shorter intervals of 3 minutes or 20 minutes were better at enhancing cortical excitability for a longer duration than the longer intervals of 3 hours or 24 hours. So we designed our second optimization study here to see if we can prolong the cortical excitability induced by our optimal protocol, which is 3 milliamp per 20 minutes, using a space stimulation technique. Here we compared our optimal protocol with a standard protocol, which is 1 milliamp per 15 minutes, which we chose because of from the literature. We compared it to sham, and we chose two intervals of two intervals of stimulation. So a short interval of 20 minutes and then a long interval of 3 hours. So just like the previous experiment I showed, we measured our baseline cortical excitability, applied our intervention, and measured the after effects immediately after stimulation, every 5 minutes up to 30 minutes, every half hour up to 2 hours, same day evening, next day morning, next day noon, and next day evening. So the results A shows the single session protocols. Result B shows the short interval, and result C shows the long interval protocols. So on the horizontal axis, we see the post measurements. We epoched the post measurements into 3 epochs: the first 30 minutes, the first 2 hours, and then the same day evening up to the next day evening. As you can see, both the optimal protocol of 3 milliamp per 20 minutes and the standard protocol of 1 milliamp per 15 minutes showed enhancements that were significant within the first 30 minutes after stimulation. The floating symbols shows comparison with sham, and then the field symbol shows comparison with baseline. There was no significant differences between the intensified protocol, that's the optimal protocol of 3 milliamp per 20 minutes, and the standard protocol. But there was a slight enhancement of the 3 milliamp per 20 minutes that was better than the 1 milliamp per 15 minutes. For the short interval results, we were able to induce after effects that was significant up to the next day evening. That is for the standard protocol. So the standard protocol after effects were still significant up to the next day evening, whereas for the optimal protocol, after effects were significant only after up to 2 hours. With a long interval stimulation, that is result number C, there was no late phase for either of the protocols. For the standard protocol, there was only a slight enhancement in cortical excitability, whereas for the optimal protocol, there was a diminution of excitability. So based on these time frames, single session anodal tDCS was inducing early phase LTP-like plasticity because it was most of the effects were gone after about 2 hours, which has been shown previously in animal studies that early phase LTP depletes quickly because of the pathway by which this plasticity occurs. Whereas the short repetition interval induce LLTP for the standard protocol and not for the optimal protocol. So induction of LLTP is considerably longer than for ELTP because of the protein synthesis and gene transcription. So the time frame is longer for this plasticity. And we believe that the short interval repetition for the 1 milliamperes showed late phase LTP-like plasticity because of the time frame. Now we propose 3 mechanisms by which this LTP-like late phase LTP-like plasticity came about. So the first one is the synaptic tagging and capture hypothesis. So according to this hypothesis, synapses that are together are able to share synaptic proteins. And by sharing these proteins, they can be able to prolongate the duration of after effects of plasticity induction. But these synaptic proteins or these synaptic tags have a half-life of about 30 minutes. So the duration or the interval between the stimulation is critically important, and this helps to efficiently recruit these synaptic tags for late phase LTP-like plasticity. We also propose that the optimal protocol was not able to induce late phase LTP-like plasticity because of the stronger effect it has on the synapses, because higher intensities are more likely to produce induce more calcium at the synapse, which can lead to some counterbalancing effects like opening up of potassium channels and decreasing the amount of plasticity induced. So you can see over here, with the right amount of calcium, and another tDCS can lead to LTP-like plasticity. But with the stronger stimulation, you are ending up in a no man's land where, according to Leisman, you are either going to get no plasticity at all, or you are going to get a reverse of LTP. Finally, we suggested that the lack of late phase LTP-like plasticity for the optimal protocol or the stronger protocol could also be a result of the synaptic modification. So according to the BCM theory, ability to induce plasticity depends critically on the state or the previous state of the network. So if the network was strongly stimulated previously, the threshold for inducing a subsequent plasticity increases very high. So you need an even stronger stimulation intensity to be able to induce plasticity. So for the standard protocol, which is 1 milliampere 15 minutes, we were able to induce early phase plasticity, but the intensity was not too much for the network, so the threshold was still low. So with the subsequent stimulation, we were able to go over the threshold and induce late phase LTP-like plasticity. But that was not possible for the more intense stimulation, because with the first application of the intense stimulation, the threshold was increased because of the high intensity. So we were not able to induce late phase LTP-like with subsequent stimulation. So in summary, we were able to expand and systematically titrate the parameter space of tDCS and show that we can induce late phase LTP-like plasticity with repeated stimulation. Single session protocols mostly induce only early phase LTP-like plasticity. But with repeated stimulation, you can induce late phase plasticity with a less intense protocol, but not with a more intense protocol. And then the interval between the first and second stimulation is critical for inducing late phase LTP-like plasticity. With a shorter interval, you can induce late phase plasticity, but with a longer interval, it is late phase plasticity induction is almost not possible. Thank you for listening.
Thank you very much, Desmond. I have time for a quick question here before maybe the more discussion questions at the end as well. Related to the 3 milliamp work that you did, do you have anything to comment on, like the tolerability and also the sham capabilities at the 3 milliamp intensity?
Perfect. So we in the paper we showed as well about the blinding as well as the side effects of the stimulation. So obviously the high intensity is more painful than the lower intensity, but if you use a topical analgesic cream, you are able to effectively blind participants during the stimulation. So based on the responses, so we had them fill out a post-stimulation questionnaire where they guessed the stimulation intensity that they received, and we analyzed that, and we found out there was no significant difference between they were not able to accurately guess the intensity that they received. And so I am assuming that if you apply these intensities without the cream, probably it will be more painful for the participants.
Okay, and so that study, all of the participants that received the higher intensity would have also had the anesthetic cream as well, yeah?
Yeah. So all the parts. So it's a repeated measure. So all the participants underwent all the conditions of So they all receive for each, even with sham, they all receive the analgesic cream, and yeah.
Great, thank you. I think then some of the other questions raised from the later part of your presentation there, I think again we'll keep those together for the end for the discussion, and there probably will be a few more questions to come back to you when we reach the end as well. Thanks, Desmond.
Okay, no problem. Thank you.
So next we have Mohsen Mosayebi to take control of the screen and show us some of his work.
So you can hear me, yeah?
Yes.
Yeah, great. Hello, everybody. Happy to have you here. But before I go into detail, I just want to ask you that we will have time till 4, yeah? The plan is to finish at around 4, yeah?
Yeah,
so in our time, yeah.
Yes.
in our time, yeah. But then
so on the system, we can overrun. So there's no time limit. There's no hard time limit. But obviously, I'm sure that we aim for about 1 hour duration for the webinars. If we need to go a bit longer for questions and things, that's fine.
Yeah. Since my my colleague also should present after me, and there should be some time, I will skip some slides. But
okay,
let's start. Okay. The tDCS clinical studies showed promising results in different to alter the symptom of different neuropsychiatric or neurological disorder. But there is a big challenge. There is a big question, and the challenge is that the overall efficacy of the technique is limited. Then what is the solution for? As I will skip these slides, because my colleague also presented. Then there is a solution, which is like manipulating the stimulation intensity and duration to find out if one of these protocols from infinite parameter space might help us to optimize the efficacy of the technique. In order to answer this question, we need a systematic titration. As my colleague explained, for anodal, here I did same. For cathodal tDCS, we systematically titrated different intensities of 1, 2, and 3 milliampere, and for each intensity, we had 15-minute, 20-minute, and 30-minute duration plus sham. And then the after effect of tDCS was evaluated using the TMS-MEP technique up to 24 hours after stimulation. Our results show that for 1 milliampere protocols, we saw typically one inhibition or reduction of corticospinal excitability about 30 minutes to 1 hour after stimulation. However, for 2 milliampere protocols, we saw no plasticity induction or plasticity enhancement, specifically for 2 milliampere 20 minutes, about 2 hours after stimulation. But for 3 milliampere protocols, again, we saw no plasticity or corticospinal excitability reduction with 3 milliampere 20 minutes. Induced longer lasting effect, or let's say optimized protocol. So in conclusion, for this titration study, we manipulated different intensity and duration. We found optimal protocol, which is 3 milliampere 20 minutes, but we also found intensity dependent effect, which is low and high tDCS intensity resulted cortical inhibition, but 2 milliampere protocol had antagonistic effect. So this package finished, and I want to present another study, the second study. tDCS have widely used to the tDCS effect, have widely used to alter also the symptom of different neuropsychiatric disorder, with the dorsolateral prefrontal cortex as the main targeted region. But much less is known for the tDCS neurophysiological effect over the DLPFC. So one might help us is TMS EEG technique to that show that that can help us to address the neuromodulatory effect of for example, tDCS or even other non-invasive brain stimulation over the different brain regions, as indexed by TMS-evoked cortical potential, or as we call it TEP, with different peaks, as indexed by TMS evoked TEPs. Okay, for over the motor cortex, anodal enhanced while cathodal reduced early TEPs, but not late TEPs. And over the prefrontal cortex, as shown here, anodal showed enhancement of early TEPs, while in another study over the prefrontal cortex, but a slightly different electrode configuration, anodal just showed a reduction of late TEPs, which is around 120 millisecond to 200 millisecond after TMS, and cathodal tDCS had no effect. Therefore, from motor to the transferability from the result from motor to prefrontal cortex, we saw some qualitatively comparable result, but the number of study is scarce, and the result also is partially heterogeneous. In order to answer this is the result and then the methodological part of the first study that I presented. We wanted to know if the result over the motor cortex, the result that we obtained over the motor cortex, is transferable over the prefrontal cortex. Therefore, we designed this study that 18 participants participated in 8 randomized sessions, that 4 sessions over the motor cortex and 4 sessions over the prefrontal cortex, including sham, low dosage, medium dosage, and high dosage of tDCS, and then the after effect probe using TMS EEG technique. Our results showed that we could recover over the both cortical region. We could recover the known TPs of early and late one using an advanced pre-processing algorithm that we developed. And for the neurophysiological effect over the tDCS, we saw that low dosage tDCS reduced the early TPs, which is P30, while medium dosage enhanced it, and then again high dosage reduced it. And over the which is in line with MEP results that we obtained, the reduction, trend-wise enhancement by medium dosage, and again reduction by high dosage. While for P60, we saw that all peaks, all dosage reduce these early peaks, but slightly after like 60 minutes. All of them are over the motor cortex. And for late peaks, which is N100 and P200, we saw no effect. Over the prefrontal cortex, in contrast to motor cortex, we saw that almost all dosage uniformly reduced with medium and high dosage with significant reduction for the early peak. And for P30 also we saw almost uniform reduction of all dosage, while again for similar to motor cortex, we did not see any significant effect on late T peaks, except perhaps for the low dosage tDCS on N100. So over the motor cortex, we saw dosage dependent nonlinear effect, which was not one-to-one transferable to the prefrontal cortex, which might be due to anatomical, pharmacological, and also functional differences. Thank you for your attention.
Thank you very much, Mohsen.
Yeah, we have enough time, kind of.
Yeah. So yeah, in the interest of time, we'll go straight through to our last speaker, Tiam Hosseinian. And then again, we'll save some questions for the end. So Tiam, I think you should now be able to share your screen with us.
Hello.
Hello. Great. Yeah.
Can you see my screen?
Yes.
Okay.
Perfect.
Okay, good.
Thank you.
Okay, good time to everyone. Thanks to Brainbox guys for invitation. I'm Tiam Hosseinian, PhD candidate from your university, and I'm working in the Department of Neuroscience and Psychology with Professor Nitsche. And my PhD project was a part of an EU project, Luminous, which was about studying, measuring, and altering consciousness by a neuromodulatory approach. And it's my pleasure to present today and sharing our knowledge. And I'm going to tell you about part of our study in this big project, which is a new technique in non-invasive brain stimulation, in short, NIBS. So, according to the previous researchers, neural oscillations are related to many higher order cognitive functions such as motor coordination, information transfer, or memory and plasticity. And also it's influenced by a vigilance level or is linked to cognitive states such as sleep and consciousness, and is associated with many neuropathological disorders like Parkinson's or epilepsy. And according to the many evidence collected so far, our question is that can we use can we use a non-invasive brain stimulation to alter the oscillatory activity directly and also alter the cognition or treat neurological disorders such as consciousness disorders and or people who are in coma, or LIS patients, and sleep disorders such as insomnia or narcolepsy. And also maybe in some cognitive deficits such as working memory. So we know that among existing NIPS techniques, rTMS can induce oscillations at a specific frequency in the brain, but the after effects are short. And on the other hand, we have tACS that can entrain endogenous oscillatory activity in the brain, but not at non-endogenous frequencies. And then this is important because in many psychiatric and neurological disorders, oscillatory activity in irrelevant frequency bands is diminished or completely absent. So maybe tACS might not be very suited. Therefore, we thought about finding a more spatial and temporal specified methods to be useful for changing rhythmic activity. And because right now there are no proper tools for that. So our hypothesis was that maybe we can be able to induce and sustain the desired narrowband frequency in the brain by combining these two techniques simultaneously together. So for this aim, we designed a preliminary study, and we built an electrical circuit where rTMS pulses could be synchronized simultaneously with tACS, and both stimulation modalities could be synchronized with each other. Otherwise, the effect from one stimulation might cancel out the effects of the other one. For this study, we had 25 participants who took part in 6 randomized sessions, and we applied 8 minutes rTMS plus tACS over the prefrontal cortex F3 and F4 EEG positions, and the stimulation block was continuous tACS with rTMS on and off, 30 seconds on and off, and we use frequency at alpha frequency at 10 Hertz, 70% AMT, and then as you can see, this 6 randomized session that we check our protocol with different protocols, rTMS alone, tACS alone, both techniques as shams, and also rTMS plus tACS at the positive peak of the tACS and also at the negative peak of the tACS, and one stimulation, both techniques as real, but we asked the participant to close their eyes during the stimulation. And we found that phase synchronized rTMS pulses to the peak of the tACS sinuses could result in a stronger effect compared to synchronization at the throat, as you can see here. And this is likely due to the fact that the electric field is inducing an excitatory effect at the positive peak, and both rTMS and tACS electric fields are aligned in the same direction. So for our second study, we were interested to apply this combined stimulation protocol for exploring causal relation between oscillation and cognition. And in this view, we looked at whether we could enhance the prefrontal theta oscillations with our combined protocol, and also if this enhancement is can be linked with improvement in working memory performance. In a first experiment, we used the same method as our preliminary study, and we applied 6 Hz rTMS plus tCS, 80% EMT for 20 minutes, and we recorded EEG before stimulation and every 10 minutes after stimulation up to 60 minutes. And then we for checking our combined protocol, also we checked this protocol with rTMS alone and tACS alone, and both techniques as shams. And for more exploration, we added 2 additional protocols. One protocol was a theta gamma, 80 Hz nested by 6 Hz. And it's a cross-frequency tACS, and which has been demonstrated to be involved in working memory. And also for another protocol, we wanted to see whether after effects could be sustained longer by intermittently continuing tACS every 5 minutes after the main block of the stimulation. So let me quickly go for the results. And as you can see here, when we compared changes in 6 Hz power to sham stimulation over the 60-minute time course of the session, the results showed that all 3 combined protocols had a significant enhancement in power in both eyes open and eyes closed, and also the grand average changes in 6 Hz power for 60 minutes. Shows in the both eyes open and eyes closed condition, the 3 combined protocols significantly enhance theta power relative to sham. And tACS only, as you can see here, showed an enhanced effect, but the effect is around 15% less. By assessing the theta power enhancement in the whole brain, we found a pattern of enhanced power in both frontal and parietal regions. And in order to assess whether these changes were functionally coupled, we measured the functional connectivity between these 2 regions, and again we found that a significant enhancement in functional connectivity with 3 combined protocols in both eyes open and eyes closed condition. And our first conclusion is that we were able to induce and sustain 10 Hz frequency over the prefrontal cortex by phase synchronized rTMS and tACS up to 30 minutes, and also 6 Hz up to 60 minutes. And also we could affect the functional network. We were able to also affect the frontoparietal network. And so we continued with our second experiment to explore whether these changes in theta frequency also translate to into functional changes in the theta frequency. In functional functional changes in cognition or not, and we use the working memory to check this. So as a second experiment, we designed like the previous one, but I mean the method, but we asked the participant to answer the n-back task before the stimulation and after the stimulation. And in our behavioral result, we found an enhancement in accuracy with measuring the D prime with our combined protocols, as you can see here, and also we saw a trend of a reduction in reaction time in our combined protocols. So our second conclusion is that with our combined stimulation, we also could affect the cognitive process and also alter and enhance the working memory. We believe that phase synchronized oscillatory stimulation could be a viable approach for further exploring causality between cognitions and oscillatory activity, and also as a therapeutic approach for clinical cases where changes in oscillatory activity are an underlying etiology, like in epilepsy, major depression, or disorder of consciousness. And at the end, I appreciate all my colleagues, family, and friends who helped me and support me in this project. And this is my email. If there is any question or comment, I will be happy to hear that. And thanks for your attention.
Excellent. Thank you very much, Tiam. And thank you to all of you for those excellent presentations as well. I'll give you a few bit of time for the questions to come in for the tDCS talk there by Tiam. Going back though to Desmond and Mohsen on the tDCS side. So do you think, based on your results, there seems to be a lot of different outcomes for tDCS, both in terms of intensity, both in terms of duration and interval between sessions? Do you think that's making it too difficult now to kind of predict the outcomes of tDCS, or do you see a potential way in your work so far that it could be possible to predict and narrow down the parameters a bit more reliably?
Desmond?
Okay, so I will answer first. So I think to begin with, these titration studies we've conducted, and then the repetition studies or the space stimulation studies are sort of one of the ways of narrowing down the parameters. So we have been able to narrow down the most important parameters for tDCS effect. These are the intensity, the duration, and the intervals of repetition. Okay, so with these studies we've conducted, we have shown that for you to induce late phase effects, you need the relatively less strong protocols. So as I showed in my presentation, you need 1 milliampere 50 minutes, or any of the relatively less stronger protocols for the induction of late phase effect if you intend to do a space stimulation protocol. But then if you're applying for single session stimulation, perhaps the relatively stronger protocols are better. So this is a successful narrowing down of all those parameters, because we cannot optimize all the parameters of tDCS. So maybe perhaps more research, we can even further narrow it down. But if I understand you correctly, you are trying to ask what is the relevance of our results. Is that a question? Asking the clinical relevance or the application of these results that we have found.
Yeah, I think that's right. You know, like that answer helps a lot, I think. And in terms of you've already narrowed down parameters and understood a lot of the parameters already. Do you think there's any more exploration and optimization to be done? And then maybe a related question then to Mohsen is about how it might be different from motor cortex to prefrontal cortex. Like you showed, in one area it might have an LTP effect, and in another area the same parameters might have an LTD effect. And is there any predictor for that, do you think? Okay.
Desmond, do you have any more comments?
No, you can.
Yeah. First, related to your first question, actually a reductionist approach that want a simple answer to this question that which parameter, let's say 1 million per 15 minutes is good for depression, and then 2 million per 20 minutes is good for a stroke and something like that. I think this is not possible, and it should be tested. We start, it's like 2-3 years that we started, even a little bit more, that we start an approach to testify different parameters, different way to see if we can optimize the tDCS protocol over specifically over the motor cortex by manipulating different intensity, duration, repetition, and we have other studies. The answer for you is that in order to find a better protocol, or let's say optimized protocol, there should be a titration study. If there is not a study yet, for example, for a stroke, you need to first titrate different protocols. There are some studies that showed, for example, 1 million per protocol, 1 million per intensity didn't work, but when they increased to 2 million per, they found that in case of Parkinson, they found significant improvement of the symptoms. But is it the case for depression? We don't know. We should test it. For the transferability, which is a big topic, yes, we showed that there is these two area is not. We couldn't find a one-to-one transferable, and then the result, it's kind of clear because the anatomical, pharmacological, and functional differences between these two areas. Therefore, the result that I showed you over the motor cortex, for sure, we showed that is not transferable. Again, if they want to use our result over the motor cortex for, I don't know, for example, for visual cortex, first they need to test it. If it doesn't work, it doesn't mean that tDCS had no effect. It means that this protocol that they selected were not well for this part of the brain or for this application. Yeah, I don't know whether I answered.
Yeah. That's really helpful. Yeah. Thank you for that.
Yeah, the general suggestion is that before saying that tDCS had no effect, you need to take a look if the design of your study was based on enough information, or it was like this a specific protocol didn't work. Yeah.
Maybe if I could put things a bit together, I think actually the results we had are not so complex. So it shows first that for anodal tDCS, with the whole range of intensities and durations we tested, we had uniform effects at the group level in the direction of excitability enhancement. Cathodal tDCS is a bit more complex because we have this enhancement with medium intensity, which is physiologically based. But we have with different intensities also excitability diminishing effects. So for cathodal tDCS, it might be a bit more important to not overstimulate. For the prefrontal cortex, I would not say it doesn't translate, but we don't see this shift. So it might be due to anatomical reasons. Reasons we have to find out now, that this missing heterogeneity, which essentially is a good message for prefrontal stimulation, where it was always discussed, for example, for depression treatment, if cathodal stimulation has also an excitability enhancement. So based on these TMS EEG results, it doesn't sound this way, doesn't look this way, then this makes things much easier. But in general, and I think that's also an important point for all these neuromodulatory techniques. So if you want to change excitability, if you want to induce plasticity, it always depends on brain state, and always has some options for non-linearities of effects. So that's a general message you have. Is also for pharmacological interventions, for example, that if you modulate, then you cannot expect that it's always linear. And if for about further optimizations, one has to take also into account that this is group results and in healthy young subjects. And then, of course, you have to look for the transferability. And then we are talking about some personalization stuff of non-invasive brain stimulation, which is also a bit more general topic, I think. But
yeah,
not the results we had, so it's not such complex.
yeah. Thank you for that, Michael. Thanks for the contextualizing that as well. And that kind of brings us on to a question back to Tiam on the tACS topic. Firstly, to clarify, is it correct that those frequencies of tACS stimulation were fixed frequencies rather than it wasn't measured from EEG, like the individualized tACS oscillations to use? Is that correct? It was a fixed 6 Hertz and a fixed 10 Hertz for those two examples?
Yeah, actually it's fixed 6 Hertz or fixed 10 Hertz because it's important for us. We didn't go for individual right now because, for example, as I told, in disorders such as consciousness disorder, people who are in coma or people who are in vegetative state, they don't have much higher, for example, frequency, and we cannot entrain the endogenous oscillatory in their brain, as they don't have anything, or it's completely absent, or it's diminished. And so we wanted to use the frequency that we wanted when there is no endogenous oscillation in the brain, then we can induce the desired frequency which we want, and then sustain that frequency to the brain.
Yeah, excellent. So do you think that's the method that you will continue with for your next studies, or is there a plan to do any individualization as well? Do you think it would be beneficial, or your explanation just now is a very good answer for reasons why not, why it wouldn't be helpful, but are there any scenarios where you think it might be?
In my point of view, I think that it's good to use the individual frequencies in the brain for any person, but not for, as I said, not for the people who are suffering from, for example, consciousness disorder, as they don't have this kind of frequency. And but for example, maybe for enhancing working memory in healthy participants, so it could be more better to use the individual frequency. I think so. Yeah.
Thank you very much. Very quickly then, I think the last question we have then before wrapping things up, and thank you again for all of your time. Again, Tiam, directed to you for this one, in terms of like the complexity of your setup, your experimental setup with the inclusion of EEG and rTMS as well. Do you think that is it just perceived complexity, or was it quite straightforward? And two, if to carry that forward to potential therapeutic or clinical applications, do you think it would be it would justify that cost and complexity, or is the reasonably similar effects that you showed with tACS only in some of the slides, in some of the experiments, would it be enough to rely only on the tACS in some cases, in your opinion?
Actually, the aim of this study was to see if we can induce and then sustain the frequency in the brain. And with tACS, we cannot induce the special frequency in the brain. We can just entrain ongoing oscillations in the brain. So in the cases that, again, we don't have non-endogenous frequencies in the brain, then tACS might not be very suited. but maybe in healthy participants, it can be helpful, but still not in the frequency, in the desired frequency that we want.
Excellent. Thank you very much for that answer as well. And once again, thank you to Michael and his team, Desmond, Mohsen, and Tiam, for your time and your presentation today. It's very much appreciated.
My pleasure.
My pleasure. Thank you.